Hi, everybody. Good evening and welcome. I'm Scotty Sims, co-founder and executive director of KCNQ2 Cure Alliance, and thanks so much for joining us tonight. This is part two of our genetic medicine webinar. In part one, we talked about RNA-targeted therapies, including antisense oligonucleotides, and that's a mouthful, also called ASOs. Tonight, we're shifting to therapies that target DNA. And Brooke Babineau, our scientific director, is going to give us a quick recap of where we left off from part one, explain how DNA targeted approaches are different, and walk us through the strategies that may be more relevant to KCNQ2 related disorders. The purpose of this webinar is just to give a simplified overview of genetic medicines that are known and which ones might be relevant to KCNQ2. A few quick housekeeping notes. As questions come to mind, just put them in the Q&A box at the bottom of Zoom rather than the chat. It just makes it easier for us to keep track of them, making sure we don't miss anything. And if you'd rather ask your question yourself, you can also use the raise the hand feature during the Q&A, and we will invite you to unmute yourself. We're recording tonight, and we'll post the recording on our YouTube channel afterwards so you can watch it again and share it with others. Our speaker tonight, again, is Dr. Brooke Babineau, our scientific director. Brooke is also an associate director of the in vivo pharmacology at Sonothera, where she leads drug development work and focuses on rare diseases. She spent much of her career studying the relationship between the brain and behavior and advancing therapies for children with neurodevelopmental disorders. And tonight she's going to help us better understand what DNA targeted therapies are, how the different approaches work, and most importantly, which ones might be relevant to our community and why. All right, Brooke, you're up.
All right. Thanks so much, Scotty. And thanks everyone for being here today. As Scotty said, a welcome questions throughout the talk. And Michelle, Scotty, and Marissa all know to just go ahead and interrupt me if they see questions come up. Sorry, questions into the Q&A, please. And they'll interrupt me and we can kind of take questions as we go. It's just me, plan to talk today. And so I really do encourage us to make it a dialogue as much as possible. And once, we get through the material, we'll definitely have time for a Q&A at the end as well. But please feel free to ask questions as we go. All right, so with that, we'll launch in. And so just the outline of today, I do want to take some time to review what we talked about last time, which really focused on the gene to protein pathway so we can understand where these therapeutic interventions are taking place. I have some new analogies that may be helpful or might just make us hungry for cookies. We'll see. And then we'll, again, review some of the RNA targeted approaches we talked about last time, and then spend some time on the DNA targeted approaches, what's different between DNA and RNA, why we have to think about delivery mechanisms and what it means to get these DNA targeted therapies to where they need to be, and the different ways you can think about DNA approaches, such as gene replacement and knockdown, and gene editing, and where these various approaches stand in relation to KCNQ2 related disease. At the very end, we also have a little bit just to, talk about genetic medicines versus small molecules. And maybe this is a fancy word for drugs or medicines that you are likely very familiar with, like your anti-seizure medicines, et cetera, just to kind of compare and contrast how different therapeutic approaches work on different parts of our biology. All right, so with that, I will launch in gene to protein pathway. So you may remember these slides from last time if you joined us last time, and if not, that's okay. We'll talk through it again. So this is a kind of a common approach for talking about the gene to protein pathway, starting with your DNA. And so when we're talking about KCNQ2 related disorders, we of course are talking about the KCNQ2 gene. The gene is something we can think of as like the level of a family cookbook. And when I talked about this last time, I failed to sort of highlight the fact that this cookbook you have to sort of think of as a sacred text. It is sitting on a shelf under perhaps a glass enclosure. It is not to be moved around. And so if you want to go and make a specific dish, you're not going to take that cookbook with you to the kitchen. Instead, you're going to rely on transcribing that recipe into a note card that you can take with you. And that note card that you can take with you, that you can think of as our RNA. The next step of DNA is RNA, with the ultimate goal to make a protein or make that dish you're going to eat. And so that handwritten copy has the notes to make your dish. Your protein is your completed dish. And to get that, you have to follow all the steps on that recipe. So again, any questions on any of this, feel free to pop them in, and my friends here will just interrupt me as we go. And so as we talked about last time, we talked about therapeutics, these ASOs, antisense oligonucleotides, that can actually work at the level of these notes to change how we get to that completed dish. We're going to review that today. And then we're also going to talk about DNA targeting medicines. And so these are therapeutics that are going to go straight to the cookbook and try and influence or integrate into the cookbook in a certain way, in which you'll sort of start from a different base recipe, such that all the pieces along the way then create a different dish, or ideally something closer to the original dish. And so again, if you remember from last time, or familiar with these biological principles, everyone has 2 copies of the KCNQ2 gene, one from each parent. So we're kind of representing a chromosome here. You get 1 copy from mom, 1 from dad, and each of these chromosomes will have a KCNQ2 gene, which is meant to be turned into a Kv7.2 protein. This Kv7.2 protein is a subunit of a potassium channel that's very important in the brain. It allows these positively charged potassium ions to flow out of our neurons and helps maintain a very important electrical balance of our brain cells, which is responsible for the communication within our brain. So as you can imagine, as we get, if this protein is not kind of cooked in the way it's meant to be, we can have impacts on the function of that channel, which impacts the function of the neuron, which of course impacts our brain and therefore behavior and many subsequent symptoms. If we go back to our analogy now of cookbook note card protein, thinking about the fact that, essentially you have 2 cookbooks for this KCNQ2 gene. You inherited one from mom, you inherited one from dad, and each of these cookbooks can be transcribed into this note card and a protein made from either one of them. And ideally, this is what the result is you get 2 identical dishes made from identical recipes. When you have a variant, and this is, the way KCNQ2 related disorders are essentially diagnosed, is based on the set of symptoms you have. Someone has sent off a genetic test. You receive that genetic report back, and it shows you have a variant, a variation. Something has changed in your cookbook for the KCNQ2 gene, right? And so that's now highlighted here by our orange stripe, right? So something is different. There's a variant in your cookbook. That variant in your cookbook is then again transcribed into your note card. You're going to copy that over, and that results in a different sort of protein being made from the original copy. Just to get a little silly with it, and because I've had an experience where my friend was making snickerdoodle cookies and I got really excited. She was visiting, and so she didn't know how we store our different resources. And instead of putting sugar in our snickerdoodles, we ended up with a salty snickerdoodle. I thought this could be a good way to think about variance and changes and what it might mean when you have a recipe swap, right? So missense variance is something that is very common in KCNQ2 related disorders, and that is something some of you might have seen in your genetic reports. This is generally a single change in the DNA sequence that leads to a completely different version of the protein being made. Okay, and so if we think of this KCNQ2 gene as a snickerdoodle recipe, so it's sitting there behind its glass enclosure, but one copy of your snickerdoodle recipe has a misprint of one ingredient. And so if one person's missense variant causes this misprint of I guess that should say salt instead of sugar, while someone else's variant might swap, cinnamon for chili powder, you're going to get very different resulting cookies, right? But in both of these cases, genetically, these are missense variants. There's just one ingredient changed, or one sort of small part of the recipe that was misprinted, and that was carried through to the making of the protein, the final product. And so that final product is different from the original recipe. And then these missense variants can result in a variety of effects on the channel itself. Sometimes this protein that's made, it's still incorporated into the channel, and potassium can go through, but it might not go through as much as it should, in which case we would consider that a loss of function. Not enough potassium is going out. But sometimes that protein change, which is incorporated into the channel, allows lots of potassium to flow out. And so that would be something we'd call it the channel level, a gain of function mutation. And we'll talk a little bit more about this moving forward. But I also wanted to introduce another consequence of these variations in the recipe, which is mislocation. This is something that's sort of newer to the KCNQ2 field, or at least in my view, but we'll be talking more about it with our research community, where it's not about how much or how little potassium is going through your channel, but it's possible that protein that was made doesn't have the right parts to keep it where it's meant to be. And so that's something we call mislocation. Instead of these proteins being placed along the axon to help this communication, they might be sort of stuck in the cell body where they're not really able to do their job. So again, there's lots of different ways for the KCNQ2 gene to be misspelled, misprinted, however you want to talk about it, and how it's sort of changed at that level of the recipe is going to impact what protein you get out at the end. I'll just pause there to see if anyone wants to interrupt or put a question in. All right. And so just moving forward to another way in which the recipe can be changed. Now I need a drink of water. Another way that the gene can be changed is sometimes the gene itself just doesn't exist. Somehow in all the merging of our genetic material that needs to happen in order to create life, some genes get deleted. Sometimes genes can get deleted, and that's fine. We didn't need 2 copies of it to begin with. And sometimes losing a gene has significant consequences. Again, if you're kind of thinking about this in the analogy of our snickerdoodle recipe, this might be a sort of a missing recipe entirely. And so if you don't have a recipe, you're not going to make those cookies. Another way you can end up with no protein being made at all from a certain cookbook is if the recipe is just jumbled in such a way that you can't decipher it in a way to make the functional protein at the end. And sometimes with either of these, if the deletion is small enough, or if the nonsense is sensical enough, you could get some version of the protein being made. And then you're kind of thinking about it again a little bit more like that missense variation that we were talking about previously, where the end product is a a different protein than the original. There's lots of other ways, that changes in the gene happen. But these are sort of the ones we wanted to highlight because tend to be the most common variants seen in KCNQ2 related disorders.
Hey, Brooke, I'm
Yeah,
just gonna interrupt and
yeah.
let you know there's a couple questions in the chat. I think the first one is more on the mislocalization or mislocation aspect. Do you have specifics of where people can learn more about this? Yeah.
Okay. Yeah, a great question. I would say for now, I would suggest reaching out to us because I don't have it off the top of my head. But if you reach out with your own variant, if you're comfortable in doing so, we are happy to check the resources we have. I will say there's probably only a handful that we maybe know for sure about this right now. And if we get a chance, maybe that's a project Michelle and I can take on of sort of um, providing a resource on the website that highlights sort of known mislocation variants. But until we have that at the ready, I'd encourage you just to reach out and we can kind of, just tell you on a one-to-one basis.
And then there is another question kind of about the language. I imagine this is my interpretation of it, about the language of loss of function, dominant negative gain of function, or mislocalization. Is mislocation a functional effect? Can something be a loss of function and have mislocalization?
Yes, that's a great question. I would consider localization, mislocation, and however we want to say that, a functional effect because it's sort of uh, it's like it's how we're talking about the protein itself, right? It's not where it's meant to be. That's sort of the consequence of the genetic mutation and the part of the protein that's changed. It somehow changed its ability to locate properly, right? So that would be a functional effect. Now your question about could you also have loss of function alongside of this, right? So now is it not in the right place and it's not letting enough potassium ions out? And I think the short answer to that is yes, I think that's wholly possible. And again, it depends on where your mutation is, what that amino acid change was, how that changes the shape of the protein and its properties, right? And I do know for sure, and again, this is something we can start to build some resources about on the website. There's a variant in which if it's and I'm not going to get the details right now. There's a variant in which it can either change. If it changes to one amino acid, you have a set of sort of functional consequences that includes mislocation and loss of function. And if it changes to a different nucleotide, it's just the loss, it's only loss of function and it doesn't have the mislocation. And so that's where I can, like, yes, you can have both effects. And sometimes it even matters just which nucleotide you change to, whether or not you have either of those features. So it's some complicated and pretty dense stuff, but we can definitely, dig into this on future calls or build some resources around it. I don't know if that answered that question, but is there anything else for now, or should we?
It says yes, thank you. But also,
Okay.
if I misinterpret your question, feel free to, I don't know, raise your hand and then ask it yourself if I do it incorrectly. I won't be offended.
Yes, happy to make this a dialogue. I usually am scared that I will ramble, but like I said to these guys earlier, I'm only taking time away from myself. So we can definitely have it more casual. All right. I will forge ahead and yeah, I think this sort of again summarizes what we kind of talked through. Um, Yeah, and so again, if we kind of look at the chromosomal picture, and so again, you have two copies of the KCNQ2 gene. Most individuals with KCNQ2 related disorders have a change or a variant in just one of those genes. So one gene might be making that, sort of original recipe KV7.2 protein, but one of the genes has some change in it that can lead to any of these sort of genetic consequences, right? Maybe there's enough of that recipe that was missing that you don't make your KV7.2, right? This is the no snickerdoodle scenario. Maybe the change in the genes resulted in this salty snickerdoodle. Maybe that means this is a dominant, this variant changed protein kind of takes over. All you can taste is the salty snickerdoodle, even when it's paired with your regular tasting snickerdoodle, right? So another word that's sometimes used in this case is dominant negative. Another way people talk about these sorts of proteins that exist, but they're changed and they kind of get in the way, is a toxic gain of function. And Lorem is a group we sort of talked about last time, and they kind of call these changes where a protein's made, but it's getting in the way of things, a toxic gain of function. And that is regardless of whether or not how that relates to our potassium channel, that is the sort of outcome of the KCNQ2 gene, right? So if we kind of look, kind of move from the gene level to the protein level and just kind of coming back up to the top in that no snickerdoodle scenario, genetically, we also might say this is a haploinsufficiency. You're missing half of the protein you're meant to be making. One of the genes is missing. You have half as much. And so in this case, you're going to have less KV7.2 made. And because there's less of these channels, you should have less potassium flowing out of the cell generally, which is also, we could consider this a loss of function at the channel level. Coming back to our salty snickerdoodle, even though it might be considered a toxic gain of function at the genetic level, that it's causing something that's going to get in the way. Again, if we think about it in terms of our potassium channel, that is our product, when that protein gets in the way, it can do one of two things. This protein can get in the way and result in less potassium flow, even less than what would be happening in a haploinsufficiency condition. And again, we might consider this a loss of function. Sometimes in papers, you might even see something like severe loss of function where that flow is really, really altered. But a different variant, a different change in that recipe, right? A spicy snickerdoodle, it might still be considered things like a dominant negative or a toxic gain of function, but this might result at the channel level with a hyperactive or overactive channel where more potassium flows out. So at the channel level, we would consider that a gain of function. So it's very, I think I said this last time, right? Like I've spent a long time academically getting trained on these things, and yet it has been very, very confusing to kind of take this genetic vocabulary and then understand it in relationship to the channel vocabulary that we use. So, if ever you have questions, feel free to reach out, because this is not straightforward stuff. Okay. Oops. And then just sort of last, I think, point related to this is that the functional effect of your KCNQ2 variant can play a role in your symptom severity. And this is important as we also start to get back into like the therapeutic piece of all of this. So that haploinsufficiency is actually tends to be what is diagnosed more as the self-limiting neonatal epilepsy. So, in all of these cases, this is presenting as a neonatal epilepsy. Again, that's often why people get to their genetic report back, is because neonatal seizures nowadays get genetic testing, and KCNQ2 is on that panel. It is actually the number 1 genetic cause of neonatal seizures. So, odds are if you have neonatal seizures, the KCNQ2 gene is more likely than any other gene to come up. But it's these dominant negatives. It's actually when you make a protein, but an altered protein that tend to result more in the developmental and epileptic encephalopathy, or the DEE. And again, this is unique to KCNQ2 because in disorders such as Dravet syndrome, it's their haploinsufficiency that is what causes the disease and causes the seizures and behavioral symptoms of Dravet syndrome. So this is important biology to understand because it actually can influence how we approach therapeutics. And so as we talked about last time, this allele-specific knockdown with antisense oligonucleotides is viable because of the fact that we can silence the KCNQ2 gene, which is making these salty or spicy snickerdoodles, and essentially turn it into haploinsufficiency, and know that is that people can sustain on a single copy of KV7.2. That, yeah, again, that's kind of unique to us, or there's other conditions that are similar, but it's really important to kind of understand that foundational biology to know how best to be able to treat the condition. So from there, we'll start to introduce some new material. And so again, we talked last time, this allele-specific knockdown approach. So it's finding an ASO that can essentially differentiate the copy of the gene that has the variant as compared to the working copy. So it's only knocking this down, leaving this guy alone. This, could work, for no matter which your cookie recipe is, if we just want to turn this off. In the case of those who have sort of the spicy snickerdoodle change, where it's that gain of function, it's actually letting more potassium out than it should. There is a theory in which you could just come down and sort of ubiquitously or non-specifically knock down any KCNQ2 you have, because if you knock that down to the right amount, you'd still have potassium flowing out through your sort of overactive channels, but maybe you can kind of titrate this such that you get the right amount of potassium flowing out of the channels that remain. This is still in what we would call preclinical or a research stage. We're trying to understand, our research individuals in our research network are trying to understand the viability of this approach. But we wanted to at least sort of mention that this is a separate ASO approach that could be specifically used for gain of function or more potassium flowing out than originally intended. Okay, so with that, just sort of talk through the differences between RNA targeted approaches and DNA. Again, feel free to throw questions in at any time. So with RNA targeted approaches, we were talking about these antisense oligonucleotides. It's important to understand they're relatively transient, and the ASO products that are on the market or in clinical trials for other disorders have sort of somewhere between monthly or 3 months, 4 months, every some interval dosing where people have to be continuously treated. The brain-related ASOs need to be delivered into the brain circulatory system, if you will, or it goes in through the lumbar through a lumbar puncture, so it can access the cerebral spinal fluid and essentially bathe the brain with this therapy. So it is, unfortunately not something at this time that can be delivered into the blood through intravenously. It's a fairly invasive delivery procedure, and again, it has to be done continuously. And as we kind of talked about, the ASO approach might change depending on the variant type and the consequence of that variant on your protein. DNA targeted approaches, some of the compare contrast here. Here we might be thinking about, again, we're thinking about targeting the gene, the original recipe. And so we could do things like replace the recipe, kind of bring a new recipe in, add a page to the cookbook, maybe, if you will, or gene editing, where like if there's that orange line that was in the cookbook, if there's a way for us to go back in and sort of paint it black, that might be how you could think about gene editing. The goal with DNA targeted approaches is to make it permanent, so it would be a single dose. The part that's sort of tricky with that means it actually at this point kind of can't redose. So if you come in too low initially, you don't have the ability to titrate up. Delivery methods are very tricky for the brain, and we're trying to get better at that. But the goal here would be to use this across a variety of genetic variants. So let's talk a bit more about that. Okay, so I think, as I said, that the DNA targeted medicines is like the idea is that they can change this original recipe. And yeah, because they are permanent, or intended to be permanent, it really it requires these special delivery vehicles so that it can not only reach the cells of the brain, but actually access the nucleus, which is where the DNA lives. And so, two different ways in which genetic medicine can be delivered are through modified adeno-associated viruses, or naturally occurring adeno-associated viruses, which are essentially dead viruses, but can still use its sort of replication potential to access the nucleus and drop something off essentially into the genetic code. Lipid nanoparticles I know a lot less about. They've been typically used more in the periphery. Baby KJ, if you're familiar with that story, a baby that got treated at CHOP for a metabolic disease where things needed to be targeted to the liver. That used lipid nanoparticles because the liver is very good at taking up lipid nanoparticles. The most commonly used delivery vehicle for the brain is something called AAV9. So just some examples is a drug called Zolgensma. That's been used for spinal muscular atrophy. You might have heard about this over the years. It's I think one of the best success stories for genetic medicine. SMA is a disease where kids were dying, before age 5 in some cases, or most cases. And this was essentially a gene replacement therapy that said, just kind of took the gene they were missing, packaged it in an AAV9, delivered it to the CSF, and now kids are surviving this, and have had very successful life trajectories. I previously worked for a company called Encoded Therapeutics, and they developed a therapy called ETX-101 that is still in clinical trials, but it's being used for Dravet syndrome. In this case, it's delivered through a neurosurgery so that a needle can be dropped into the space in our brain called the ventricles, where the CSF lives in the brain. And when that AAV9 therapeutic is dropped in there, they can essentially upregulate the amount of SCN1A that's produced by the working copy of the gene in Dravet patients. And they've been seeing some really good preliminary results as far as seizure reduction and even some behavioral improvements. I'll just pause for a second. I didn't know if there was some activity yet.
Yes, there are some questions in the chat. One question is requesting to be answered in Spanish. I'm not sure if you can do that, but we can maybe answer it in English and then find someone who might be able to translate for us, and then reach out via email. But the general question is, does a gene therapy exist for KCNQ2 related disorders, and is it likely that that will become an option?
Right. Unfortunately, despite my 20 plus years in California, I have not become fluent enough in Spanish to answer in Spanish, but we can 100% get some of these answers translated and sent back out. I took German in high school. It doesn't feel very useful at all, but anyway. So does a gene therapy exist for KCNQ2? Was that the basis of the question? Okay. There's a uh, couple ways for me to answer this. We are aware of a individual in China who has used the mechanisms available there to test a experimental gene therapy in some number of patients. That is sort of the most or we know about that at the moment. That researcher will be speaking at our research roundtable in September. That's sort of our gathering of our research network. So we, will better understand sort of where that is and what will come of it, I think, in September. Beyond that, there are some therapies in development, and I'll sort of talk more through them as we go through the rest of the slides. Is there
Brooke,
anything
there
else?
was
Right?
another
Yep.
question.
Mhm.
It says, is there a reason you can't titrate up the dose with lipid nanoparticles? They understand the reason you can't do multiple dosing with viruses is the immune response.
Yeah, with lipid nanoparticles, I actually think you can. And again, I'm less of an expert there. I think, yeah, right now the limitation for us in lipid nanoparticles is that they don't, there's not that, to my knowledge, a lipid nanoparticle that can cross the blood-brain barrier. So for brain-based genetic medicine, right now it's been primarily AAVs, but again, don't quote me on that. There's probably someone out there working on something that I'm not aware of. But yes, with lipid nanoparticles, I think, even going back to baby KJ, I think he had multiple doses of LNPs. And so there's definitely room for titration there. I will also just use this opportunity to mention, so Sonathera, the company I work for, is actually trying to use ultrasound to deliver genetic medicines. Unfortunately, we're not quite ready to work on the brain yet, but there's a lot of work in delivery and trying to get away from viral mediated, so you have this flexibility of being able to find the right dose. Anything else right now, or push on? Okay. Okay. And actually the previous question was kind of a good segue here. So as I mentioned, we are aware of an individual in China. Their regulatory system is different from ours, and there's a way in which individual researchers can initiate very small. It's kind of like an N of one, trial system that we have here. So that's essentially what's happening is they're using a gene replacement strategy, which I'll kind of talk through here. Which is essentially delivering a brand new copy of KCNQ2 to help supplement the production of the working copies of KV7. So again, if you think about these dark purple, chromosomes, sort of um, being your sort of biological material, the idea of a gene replacement therapy is to essentially deliver an additional KCNQ2 gene, and then even though you might still be making the variant protein, the hope is that this additional working copy might dilute the consequences of that gene. I think where it's more clearly going to be beneficial is going to be if in the case of a haploinsufficiency, right? If you're just not making that protein, well, then delivering another gene and having that gene supplement the lack of the KCNQ2 gene is more straightforward, biologically to see the benefits of. And so the strategy, again, currently used in China is an AAV9 based therapy like ETX-101. It is delivered directly to the brain via a neurosurgery. We hope to have updates for the community later this year. Then another strategy, if you kind of build on this and kind of think about how you can kind of make it potentially more beneficial for those cases where you are making this protein that's getting in the way, you can think about a gene replacement plus knockdown approach. And so again, the idea with these ASOs essentially is to kind of turn off the gene in some way. And so what's tricky about the cases where you have the KCNQ2 gene that leads to less potassium flowing out is that you wouldn't want to touch your working copy of KCNQ2, right? But if you are going to go ahead and replace KCNQ2, you can kind of more ubiquitously just knock down the KCNQ2 you have endogenously or inside yourself. And they can use this gene replacement to kind of, bring enough KCNQ2 to kind of overcome the knockdown that's been created. The benefit of this is it should work for any variant type. And there's a company called Asymptote Genetic Medicine. They came to our last summit and gave a talk. They are working on a strategy just like this. And it's even a bit more complicated than what we're even showing, where they can have some pieces, additional pieces built into this whole genetic package that can kind of tell the gene just how much KCNQ2 to produce. So essentially you can get to physiologically relevant or the right amount of KCNQ2 by having this sort of special genetic sequence that can tell you when the right amount has been achieved. So it's a pretty sophisticated therapy. It's very exciting that this group is working on it, and we hope to continue to support them in any way we can to move this forward. Then the last thing I'll touch on in sort of the genetic medicine space is this idea of gene editing. You might also hear this as CRISPR, prime editing, base editing. These are some of the terms around gene editing as a broad category. And I won't get into the specifics here today, but the idea is that you could cut into the DNA to make changes to the variant copy of the KCNQ2 gene. And essentially just, in that way in your genetic report, you might have seen that your C was turned into a T. This could just switch it back, change that T back to a C. You now essentially have that original recipe back in your cookbook, which should then lead to the original protein being made. This is in very early development stages for brain-related disorders, again, largely due to the fact that, delivery is a challenge. And I will say too, I think, my view of gene editing is there are efficiency challenges that you can definitely make the changes of the gene, but it's often in only a % of cells. And so like if you go in with a dose with your CRISPR and see how many cells did I change, it might be 10% or 20%. And so again, this was sort of the mechanism used for baby KJ, but they were able to go in and give more. And each time they gave more cells, changed over to the working copy. And I just don't think we're quite in a place with brain delivery in which we can kind of be able to sort of titrate up and make sure we change the right amount of genes. But this is an important technology to be informed on, and we are working to have the right research partners so that some KCNQ2 gene editing work can be done. And I will also say, credit Kara for actually initiating some efforts with NYU. Kara Bolter, who spoke last time, for initiating some efforts at NYU around gene editing. And we hope to sort of carry that forward to other researchers who are kind of working in the gene editing and channelopathy space. Okay. And so just to kind of summarize all the different ways in which we might think about genetic medicine, starting from the top, we can think about reducing the variant copy of KCNQ2. This would be targeting the RNA And we talked last time about programs at NLorem and Boston Children's Hospital that are working on this allele-specific silencing approach. Theoretically, this should work on variants that either result at a channel level in gain of function or loss of function. Essentially, if that protein is dominant negative or getting in the way, then it would be appropriate to think about turning that off specifically and relying on that working copy of KCNQ2. Another idea, depending on the effect, could be just reducing all copies of KCNQ2 to a certain level. Obviously, we still need some Kv7.2 made to let those potassium ions out. This would be an RNA target. Some of our research collaborators are working on this. This would be a non-specific silencing of using an ASO, but this would only work if your channels are otherwise letting more potassium out than they should be. Then if we start to think about DNA, we talked about the idea of a gene replacement or adding a new copy of KCNQ2. This is a program in development slash, it is technically, in people in China. But it's sort of how it will progress from there is a little bit uncertain. Again, we consider this a gene replacement approach, and this we think would have, biological relevance for individuals who have a haploinsufficiency, but it's possible it can also overcome some of the dominant negative variants. And then the asymptote genetic medicine approach of reducing the innate expression of KCNQ2 and replace it with a new copy. That's also a DNA targeted approach. Can consider this a knockdown and replace. And ideally, this could be beneficial to any type of mutation, and so a very ubiquitous therapy. And then the idea to specifically edit KCNQ2 variants. Fixing that innate copy, again, DNA targeted. This is very much in a research level stage, but ideally this could fix any variant. So it could also be a ubiquitous therapy, but I think still a few steps away from some of the other ones listed above it.
Brooke, there's another question.
Yeah.
It's about the Boston Children's Hospital work. Is there a certain age range that they're looking at to use their RNA approach with, or is it kind of for anyone?
Yeah, that's a good question. And I don't know. They did a webinar, and I'm trying to remember if age came up. I don't know if Michelle or anyone else remembers. I don't think there's an age range for them just yet. I know they want to kind of move towards clinical trials, but they have not, I have not yet seen any, kind of clinical trial criteria, or don't recall them having any specifics just yet. But is anyone else? Yeah. But I think,
Okay.
I think, obviously we're in touch with them as they get closer to clinic, we can let people know.
And then there's another question if there's anything happening in the UK regarding these kinds of therapies, or are these all US based?
So, and NLorem right now, I think is US based. I can't remember if there's something about like you can apply if you're not US and that you'd have to get yourself here and get treated in the US. If anyone who has, if anyone's online who has experience with that and NLorem and wants to, express anything in chat, please feel free. But what I will say is like, asymptote genetic medicine, they're a biotech, right? Like they will be looking at any space where they all have the opportunity to treat. So, with encoded, like ETX101, like, yeah, they, as a company, you sort of choose which regulatory bodies you want to go through to try and get approval. And so, yeah, encoded went to Australia, UK, I think even before the US. So I think, as some of this develops, we'll have a better sense of where it might become available. But with with NLorem and BCH, I think those are sort of treatments in the US, but it doesn't mean that it's not available to those who would be willing or able to travel to the US for treatment. Okay, anything else on that, or just sort of move on to just this last part. Just to want to just kind of make the comparison, to these genetic medicines, which we just talked about, trying to go and manipulate the gene or the, sort of the adjacent piece of the gene, this RNA, to sort of change the recipe, change the note card so that which will lead to that completed dish. But as many of you are probably very aware of, like the drugs are out there that work on proteins, right? I sometimes call them small molecules, but these are the drugs, the medicines. And so while right now there's not drugs that are targeted to KV7. There has been, and this was a drug called retigabine. And this is something that can work on the channel and sort of influence the amount of potassium that can go through. And there is, what that would look like is just sort of like kind of looking at a cell, and these are our KV7 potassium channels. KV7 can sometimes make a channel with KV7.2 can make channels with KV7.3, or it can make channels with itself. But again, if there's the idea for these potassium channels is that you let potassium ions from inside the cell to go out. And there are drugs being made that can interact with these channels in such a way that it can increase the amount of potassium ions flowing out. So for those individuals with mutations that restrict the amount of potassium flowing out, this could be a targeted therapy that does help the sort of functional consequence of all these genetic, changes along the way. So, I think also stay tuned for the fact that there are drugs in development that will also act on the channel. But again, the drugs that are farthest along are these potassium channel activators. So these would not necessarily be appropriate for those who have gain of function mutations or mutations where already there is too much potassium flowing out. So I think that is really why, it drives us to still, to look into these genetic medicines therapies and really try to cover the gamut of variations and the functional consequences that individuals with KCNQ2 related disorders face. Okay, I think that brings us to the end. And so I thank you so much for asking questions along the way. It really helps break things up. And so I don't feel like I'm speaking into a void, but I am happy now to take questions on anything we didn't cover or any just burning questions you'd like to ask.
You got a question in the Q&A section asking if a potassium channel opener could improve other outcomes like cognition and speech beyond seizures. Is that a possibility?
Um, Short answer, yes. I think it's a possibility. If it really can kind of rebalance the electrical activity of the neurons, like that would be the idea that it can, it is being trialed as an anti-seizure medicine, the drugs that are in clinical development. But I think in the case of, KCNQ2 related diseases, I think there's a real chance that it can have cognitive benefits. Scotty, do you want to, talk about your experience with ortigabine at all, or
Yeah,
just give
I
your
can.
perspective? Yeah.
Yeah, I can certainly do that. Our daughter tried retigabine. She's 15 now and tried it when she was 14 months old. Um, and we did see cognitive and developmental improvements. At the time, she was seizure free, not on anti-seizure medications. And so we really did a trial to see if we could see any kind of developmental improvements. And we did. They were small, but we did see them. So I think there is definitely the chance that you can, and hope that our children can respond in that way as these medications become available. And Brooke, I also received a another question asking about is it a possibility to do a combination of treatments?
Mm. Yeah, that's a great question. And I feel like I've talked with Kara a bit about this as well. Yeah, theoretically, um, you can be on an ASO and have an AAV. And I say theoretically because, biologically, there's no reason you couldn't do those things. If we get down to like a clinical trial and whether there's exclusion criteria based on what you're on, it could be a different story, but I don't know that we're not at that place yet where we're, have sort of clinical trial requirements to be up against. But yeah, biologically, there's no reason you couldn't be on an ASO and receive a gene therapy. Um, I'm just trying to think about if there's any one or the other. Like, yeah, I mean, if you're on an ASO, allele selective ASO, and a gene replacement gene therapy was available. Again, biologically, there's no reason you couldn't do both, and you could definitely imagine therapeutic benefits from combining them. When it comes to like small molecules and ASOs, again, I think biologically, that could totally work together, and I think in some ways that could be beneficial. You can imagine again if you're on this allele selective ASO, you've knocked down the copy that has the variant, and you put on a KV7 activator because essentially if you're creating a haploinsufficiency case for yourself, maybe you still want to increase the flow of potassium ions through the channels that are made with the one working copy, right? So yes, biologically that will make sense, and then I think again, if you're thinking about clinical trials and what their requirements are going to be, that might be different. But biologically that should work as well. Any new questions?
Yeah, there's one in the chat talking about kind of if you're not sure if your variants gain of function, loss of function, kind of like
Yeah.
classification, what are the next steps to work with researchers to determine that?
Yeah, great question. Because unfortunately there isn't a great system to know that right now, right? Like this, it's likely that's something that doesn't, get reported out on your genetic report because it's not something that's necessarily available clinically. It is sort of a research-based characterization. So we are working to have resources that show the published characterizations, and so you could go and look at that. I can't actually remember if we've put it out on the website yet or if it's still waiting review from some of our research network. So yeah, again, you can start by asking us and we can go to our resources, but I will sort of, I guess, caution that unfortunately there's still a lot of variants that are unknown. Um, and so we are definitely thinking about not just us, but a lot of other channelopathies, like how to kind of harmonize the system, if you will, so that more characterization is done and more variants are their functional consequences known, because obviously this can influence, what medications and what therapeutic approaches are right for you.
Well, Brooke, thank you so much. I mean, that was really helpful. And I think it's an ever changing landscape, and making that digestible and relatable for our community is great. Want to thank everybody for joining. And I don't know about you, but I will be baking some sweet, not salty snickerdoodles this weekend. Um, so yeah, and always know that you can reach out to us if you have an idea for a webinar or a topic you would like to see us address. We're very much open to suggestions on any topics. Thank you, everyone, and have a good evening.
Great. Thanks so much.